Hello, Mr. P here. This lecture is Chapter 7, Lecture 4, and we're going to talk a little bit about endomembrane organelles, which are specifically the nucleus, the ribosomes, endoplasmic reticulum, both smooth and rough, the Golgi apparatus, lysosome, and vacuoles, and may even talk a little bit about the cell wall if we have time. So when we talk about endomembrane organelles, we're specifically talking about the organelles that are inside of a eukaryotic cell. Remember, prokaryotic cells do not have a nucleus, do not have a membrane around their DNA, and don't have any of these things except for the ribosomes. Remember, the ribosomes in a prokaryotic cell are 70S. The ribosomes in a eukaryotic cell are now 80S, and we can differentiate the difference in class. But let's talk a little bit about the organelles. So nucleus. Nucleus you probably recall as being the control center of the cell. It contains all the inheritable DNA, and specifically the DNA is within a membrane called the nuclear membrane, or sometimes you'll hear it be called the nuclear envelope. And one of the important things to know about the nuclear membrane is that it's porous, and basically, like anything that's porous, it has holes, and those holes allow things to go in and out. Specifically, RNA that is made in the nucleus has to leave, molecules have to come in, proteins that are made by RNA and DNA that are in charge of DNA replication and enzymes have to come in and out. And all that stuff is going to be talked about in detail in Chapter 12. But for now, know that the nuclear membrane is a double membrane that is surrounding the DNA, but it has to be porous because we have to allow certain things to go in and out. Just like the cell membrane is semipermeable, the nuclear membrane is semipermeable as well. Okay, the nucleolus is the core of the nucleus, and it's the place where ribosomes are made. It's also the place where the RNA is made. So if you were to look at the picture in the next slide, there is a kind of a dense ball of DNA in the core of the nucleus. That core is called the nucleolus, and that's where all the DNA is stored, and that DNA is stored in the form of chromatin. Okay. On the outside of a nucleus is a nuclear envelope, and here's a picture. Okay. Here is the thick, kind of dense ball of DNA called the nucleolus. It is a ball of DNA. That DNA is in the chromatin form, and that just means it's kind of a loosely packed, non-specialized, non-organized way of packaging DNA. On the outside of all this DNA and the outside of the nucleus is a nuclear envelope, and it's a double membrane or two-layer. You can see two layers if you look really closely. And through the two-layer membrane are these holes, and these holes are called nuclear pores. Those pores allow RNA that is made here to leave. It also allows proteins and enzymes to go in, because those proteins are going to be instrumental in the replication and transcription of DNA and RNA. The nucleus is always attached with this little channel right here to the rough ER, okay, the rough ER, endoplasmic reticulum. And the rough ER looks rough because it is studded with these ribosomes. Remember, the 80S eukaryotic ribosomes. And ribosomes are in charge of making proteins. And so if the ribosomes, which are in charge of making proteins, are all over these folded up membranes, what do you think the job of the ER is? I would hope you would suggest that the ER uses these ribosomes to produce and synthesize proteins. Okay, produce and synthesize proteins. Really important. Why do you suggest that these membranes are folded up like this? Okay, pause it, think about it. Okay, so the membranes are folded up like this to maximize surface area and minimize volume, because what happens at the surface of membranes? Magic happens at the surface of membranes, and the magic is in the form of protein production. Okay. Ribosomes. So if we talk a little bit about ribosomes, I kind of just turned the nucleus a little bit. Again, we see that the membranes that make up the nuclear envelope of this nucleus are consistent with the membranes that make up the ER, specifically the rough ER. And again, it's rough ER because it appears rough. When you look at the rough ER under a microscope, it literally looks like it has all these dots on it, and all those dots are ribosomes. And the job of the ribosomes is to produce proteins. Okay, so what is the job of ribosomes? Again, produce proteins. The ribosomes can either be attached to the rough ER or they can be free in the cytoplasm. But if they are free in the cytoplasm, they have to be attached or anchored to the cytoskeleton because they have to have something to adhere themselves to. And how do the proteins make themselves, or how does the ribosome make the proteins? They are going to convert an mRNA message into protein by the process of translation. And again, we'll learn about that more in detail in chapter 12. But for now, we need to know that ribosomes produce proteins by taking mRNA messages, reading them like a recipe, and producing proteins by placing amino acids together. And if you remember from chapter 12, or chapter 2 rather, that placing amino acids together is building polymers from monomers, and that process is dehydration synthesis. So don't forget about your chapter 2 stuff. Okay, we're just building on the things that you learned in prior chapters. The endoplasmic reticulum, if I take the nucleus out of this picture and just keep the nuclear envelope, it's continuous with the ER, and notice that all this rough ER looks rough because it has the ribosomes on it, but over here now we have these tubular membranes, and those tubular membranes are the smooth ER. Now, structure dictates function, and so it makes sense that if these membranes are structured well in the production of proteins, that it makes sense that the smooth ER's job is not to produce proteins, or they would be structured the exact same way. In fact, the smooth ER's job is to produce lipids. Are lipids the same as proteins? No. And so if they're producing something that's different, you would expect them to be shaped differently. Here's an actual electron micrograph, and you can see here that when you're looking at rough ER, just like this, it's folded up cellular membranes, and it has all these dots. All those dots are ribosomes, and then when you get to the smooth ER, they're more tubular, rounded, kind of abstract. They're not just nice, even rows of ribosome-rich membrane. Okay, so the job of the ER is or the structure of the ER is a network of flattened sacs, and all of those interconnected sacs lead to the production of the ER, and there's two types, the rough ER and the smooth ER. We've already talked about what the rough ER and smooth ER do. Rough ER produces proteins, smooth ER produces lipids. They both are continuous, and they're one continuous organelle. Okay, so the rough ER, just as a recap, studded with ribosomes, their job is to produce proteins, and some of those proteins can be enzymes, some of those proteins can be antibodies, some of those proteins can be antigens, all kinds of things. Okay, the rough ER also rearranges proteins and adds chemicals to other proteins in the ER, meaning some of the chemical modification of those proteins are going to take place in the rough ER. After the proteins are produced and modified briefly in the rough ER, they are going to be transferred in vesicles to the Golgi apparatus, where the final modification and the final production into the vesicle happens. Okay, the Golgi apparatus is kind of like a post office. Smooth ER, if we kind of talk about how it's different and similar to the rough ER, the smooth ER, remember, is continuous with the rough ER, although the ribosomes stop. It's more tubular shape, and they are in charge of producing lipids or synthesizing lipids, and both types, phospholipids and triglycerides, are produced by the smooth ER, and those lipids are used for a variety of tasks in the cell, meaning we use the phospholipids. Remember this molecule that looks like this? The task of that lipid is to make biological membranes. The task of this lipid is to produce long-term energy storage molecules. Okay, so those two types of lipids are produced by the smooth ER. And The smoothie R is really good about storing calcium ions, and those are necessary for muscle contraction. You could not go out and play a sport without the lipids that are produced by the smoothie R or the ions that are stored by the smoothie R as well. So we talked a little bit about Golgi apparatus already. The Golgi apparatus is a structure that looks like a stack of flattened membranes, but they're different than the rough ER because they're not connected. Now, if we were to compare, the rough ER, remember, looks like this. It's a bunch of connected membranes that kind of go back and forth and have all these little dots on them. Those dots would be ribosomes. The Golgi apparatus looks like a bunch of flattened sacs, but see how these are all different and not interconnected. Okay, so there's the difference. They look similar. The rough ER is all interconnected. The Golgi apparatus is not interconnected. Not interconnected is important. The Golgi apparatus is going to receive and modify proteins from the rough ER, and they receive those proteins in the form of vesicles. The Golgi apparatus' ultimate job, though, is just to transport the proteins that they receive from the ER into vesicles, or put the proteins into vesicles that are made from the lipids that are produced by the smoothie ER, and then transport them into and out of the cell and all over the place. Okay, it's kind of like I said, the post office. And there you go. Acts like the post office. Here's a picture of the Golgi apparatus. There is always a receiving side of the Golgi apparatus, and there's a shipping side of the apparatus. And these vesicles will move in one direction from the receiving side to the shipping side. So when the Golgi apparatus receives these proteins from the ER in the form of a vesicle, it's going to go from this sac to this sac to this sac to this sac, and then ultimately out in the form of a transport vesicle. After it's been modified, after it's been basically chemically altered, it's going to be packaged and reshipped with the proper labels. Vesicles are those lipid-bound bubbles that we're going to send materials to and from the cell, and that's going to lead into our bulk transport lecture, which is going to be at the end of this chapter, probably lecture 9. But vesicles are small membrane-bound sacs that divide some materials from the rest of the cytoplasm. This all would be cytoplasm out here, and whatever we want to keep in there free from the cytoplasm is going to be transported by these vesicles. Okay. It's just like a delivery truck, right? These extracellular food particles are going to come in, they're going to pinch in our cell membrane. These, once this membrane kind of folds in on itself, it's going to completely adhere, creating this bubble, and they're going to come into the cell, or they're going to go from the Golgi and they're going to go release the material out of the cell. That would be exocytosis. So here is a lysosome. Lyse. Lyse is a prefix to mean, or means to split or break down, and so these lysosomes are special types of vesicles that are in charge of breaking down bad things, whether they're bacteria cells or maybe not so bad things like food and water. But they're basically bubbles that contain digestive enzymes. And so if we follow the entire production of a protein, the ribosomes that are attached to a rough ER are going to produce a protein and are going to send those protein in a transport vesicle to the Golgi. Now notice these are inactive enzymes at this time. Okay. The inactive enzymes are going to be bound to the first sac on the receiving side of this Golgi. They're going to transport between sacs eventually to the shipping side. Once they are shipped out, they are completely activated. And once these enzymes are activated in these special sacs called lysosomes, they will meet and join themselves with a vacuole or a vesicle that's containing food, water, or bacteria cells, and will be used to digest this stuff, and then will release it into the cell so the cell can eat. Okay. So they're Greek for breakdown body. They contain hydrolytic enzymes for material breakdown and for digestion of materials. destroys the cell when it dies. And so when the cell dies, like all living things do, these enzymes will be released from these lysosomes and basically digest the cell from within. Vacuoles. Vacuoles are storage containers that contain water or food. When you look at a plant cell, you always see this large void, and it looks like nothing's there. But in reality, that is a central vacuole that contains water. And water is essential for cellular function. And plant cells have bigger central vacuoles, because they are not motile, meaning the vacuoles have to expand and hold more water because the plants are basically at the mercy of the environment and will only refill these vacuoles when it rains or when there's sufficient groundwater. Okay, animals, when they get thirsty, they go find water, and so animals don't have to store as much water in their cells. But these vacuoles are short-term storage places for the cell. They store food, water, and waste, all in different vacuoles. Okay, it's kind of gross to think that they would store food, waste, and water all in the same bubble. They don't. Okay, they have different vacuoles for each. Plant cells have very large vacuoles. I already stated that, right? Large vacuoles because they are not able to go find water when they're thirsty. Centrioles. These centrioles are structures kind of like rigatoni noodles. They kind of look like that. They're made up of microtubule triplets, and all in all, there's 8 of them around this core, and their job is to basically pull the cell apart when it's going through cell division. And so when we talk about cell replication, like mitosis or meiosis or binary fission, these microtubules are going to be arranged at the poles, or these centrioles are going to be at the poles, and they're going to produce microtubules that are going to extend from the poles into the equator of a cell and eventually pull the entire cell apart. The cell wall is in charge of the protection and support for plant cells or insect cells or fungus cells. Plant cell walls are not they're not in charge of regulation of materials, right? They allow materials to pass only through openings and or channels. The cell membrane is really what regulates the materials. The cell wall is a rigid layer that provides protection, support, and shape to a cell. There is a reason why plant cells are harder and more rigid than animal cells. It's because of the cell wall. Okay, they're made of cellulose in plants, chitin in insect and fungus cells, or peptidoglycan in bacteria cells. Okay, all 3 of those are complex carbohydrates, but they have a slightly different structure, and so they have a slightly different function. That's all that's going to do it for the endomembrane organelles. Stay tuned. See ya.